Hello, my dear friends. Welcome to the new session of Pharmacognosy. Myself, Miss Malvi Rajendra Bhavsar, M.Pharm in Pharmaceutics. So today we will start with Pharmaceutical Aids, Part 1. In this we will study honey and arachis oil, kaolin and guar gum. After watching this, students will be able to learn what is pharmaceutical aid, different crude drug uses pharmaceutical aids, and monograph of crude drug. So first we'll start with pharmaceutical aids. Pharmaceutical aids definition: the substance which are of little or no therapeutic value but are essentially used in manufacture or compounding of various pharmaceuticals are known as pharmaceutical aids or pharmaceutics necessity. Sources of pharmaceutical aids. First source is animal source. Example of animal source is beeswax and lanolin. From vegetable source, acacia and fragacanth. Mineral source, it is kaolin. Then pharmaceutical aids classification. So according to color, example are saffron, turmeric and indigo. Diluent, cinnamon water, peppermint oil. Disintegrating agent, example are starch and CMC. Emulsifying and suspending agent, example are acacia, agar and gelatin. Flavors, example is rose, lemon oil, orange peel. Lubricant, talc, cocoa butter. Ointment base, beeswax, wool fat. Solvent, example is alcohol and glycerin. Sweetening agent, example is glycerides and honey. So first pharmaceutical aid we are going to start is honey. Synonym is madhu or male. Biological source, it is a sugary secretion deposited in honeycomb by the bees Apis mellifica, Apis dorsata, and other species of Apis, family Apidae. Geographical sources Africa, Australia, New Zealand, and California and India. Description of honey. Color, it is pale yellow to yellowish brown. Odor is characteristic and pleasant. Taste is sweet and faintly acrid. Solubility is soluble in water, insoluble in alcohol. Then some standards of honey. Total ash is 0.1 to 0.8%. Weight per ml 1.35 to 1.36 gram. Specific rotation is plus 3 degree to minus 10 degree. Chemical constituents, it contains 35% glucose, 45% fructose and 2% sucrose. It also contains dextrine, formic acid and other enzymes. Other chemical constituents are succinic acid and gums. Then uses of honey. Demulcent. It acts as demulcent, sweetening agent, antiseptic, vehicle for Ayurvedic formulation. Then storage condition of honey. It should be stored in well closed container, protected from light and in cool and dry place. Then next is arachis oil. Synonym groundnut oil and peanut oil. Biological source, fixed oil expressed from seeds of Arachis hypogea, family Leguminosae. Geographical source, Brazil, Southern United States, China, Africa, India. The description, color of oil is pale yellow, colored liquid. Odor is faint and characteristic. Taste is bland and nut-like. Solubility is soluble in alcohol, solvent ether, chloroform. Chemical constituent are glycerides of fats acid. Chiefly, it contains oleic acid, linoleic acid, stearic acid, arachidic acid, lignoceric acid, palmitic acid. Uses of arachis oil: solvent for intramuscular injection, in preparation of liniments, plaster, soaps, ointment, cream, lubricant, edible oil. Then comes our third, kaolin. It is insoluble in water and in mineral acid. It absorbs small amount of water, hygroscopic in nature. Identification test of kaolin on charcoal block with cobalt nitrate. A blue mass is formed due to presence of alumina. Uses of kaolin: adsorbent in treatment of enteritis, colitis, dysentery, and alcohol food poisoning. It is used as externally as dusting powder, clarifying agent during filtration. Then other uses are it in anti-caking preparations, cosmetic, insecticide, and paint. Source of alumina, ingredient of toilet powders, base of disinfectant powder. Then fourth pharmaceutical aid is guar gum. Synonym is guar flower or jaguar gum. Biological source: gum is a powder of endosperm of seeds of Cymopsis tetragonobulbus, family Leguminosae. Geographical sources: Gujarat, Maharashtra, Karnataka, Rajasthan, Pakistan. Then description of guar gum: color: it is colorless and pale yellowish white color powder. Odor is characteristic. Taste is gummy and characteristic. Solubility: dispersible in water forming colloidal solution. Insoluble in alcohol. Identification test: first is guar gum with ruthenium red. There is no pink color. Then guar gum with 2% lead acetate, it forms precipitate. Guar gum plus weak solution of iodine, no olive green color is obtained. Then chemical constitution of guar gum: contents are divided into water soluble and water insoluble part. Water soluble fraction consists about 85% of the gum, which is known as guaran, which is hydrocolloidal polysaccharide. Guaran or hydrolysis yield 35% of galactose and 65% mannose. Guar gum also contains 5 to 7% protein. Uses: thickening agent, it has 5 to 8 times more power than starch. Protective colloid, binding and disintegrating agent, bulk laxative. It is also used as appetite depressant in peptic ulcer therapy, emulsifying agent in paper printing, polishing and textile industry, in food and cosmetic industry. So in this we learnt about monograph of honey, arachis oil, kaolin and guar gum with their uses, chemical constituent and biological source. Thank you very much for attending this session. Next pharmaceutical aid will be seeing in next session. Thank you. Thank you very much, friends.